Okay, good afternoon, everybody. And this afternoon, we're going to be discussing forensic serology, which leads on quite naturally from all of our discussion about drugs and about toxicology. Serology is actually the study of human blood and of human blood products and of body fluids. And indeed, sometimes toxicologists are called on to perform forensic serology tests in order to discern whether drugs are present in the blood, etc. Blood has for a very, very long time produced extremely important forensic evidence. And we need to understand a little bit about blood and about how it works. One of the forensic features of blood that you will have heard of course, are the blood groups. And in the early 20th century, when blood groups had been clearly defined and discerned, it became a standard procedure to examine blood at the scene of a crime to try to discern the blood group which it belonged to. In other words, the blood group of the person who had produced the blood. This can produce very important class evidence. It does not produce individual evidence, but it produces class evidence. And very frequently, its real importance was to exclude people rather than to include them in a pool of suspects. In any case, blood groupings are still done. It's still an important forensic procedure. And it not only in criminal situations, but in questions of paternity and things like this as well. Blood group may be a very important piece of evidence. So we need to understand a little bit about blood, about what it's made up of, and how these systems work. So if you take a sample of blood, this is what it looks like. Obviously familiar to all of us, this opaque red, deep red color. The red color is conferred by the hemoglobin in the blood, which is contained in red blood corpuscles, red blood cells. If you put this into a centrifuge and you spin it down, you can spin down all of the heavier blood cells and leave clear plasma. This is representative of the fluid that makes up the bulk of the blood. It's about 55% of the blood volume is this clear fluid. The fluid is full of proteins of various sorts, salts, ions of various kinds. If we look carefully at this plug of blood cells at the bottom here, well, first of all, see that there's this very big plug here, which are the red blood cells, the erythrocytes. They are the commonest and form the bulk of the blood cells that are in circulation in the blood circulatory system. But look carefully here, and you'll see that there's a layer here of white blood cells. And white blood cells become very important in forensic science because white blood cells have a nucleus. Red blood cells do not. But white blood cells contain DNA, which can be typed to an individual. The red blood corpuscles do not have a nucleus, but they do have some very useful features which can be used to characterize them. There's also, by the way, contained in this layer here, there are some very, very small cells called the platelets. They almost like fragments of red blood cells in a way. They have no nucleus. Very important because they are responsible for causing blood to clot. They're called the platelets. They're present here. No DNA in them. If you want to do DNA typing, this is what you're particularly interested in, are the leukocytes or white blood cells. Okay, so all of the features of blood grouping are associated with characters of the erythrocytes, of the red blood corpuscles. So the red blood corpuscles around the outside of the corpuscle, they have a membrane. And that is the membrane which defines the cell itself. And embedded in the membrane are many, many proteins and other substances, glycoproteins, all sorts of things. And there's one particular group of these molecules which are of particular significance. And these are the antigens which confer character on the blood corpuscles. Now, an antigen is any substance which can excite a reaction from the immune system. But there's a rule. Our cells are made up of proteins, all sorts of different molecules, which could excite an immune reaction. But of course, we can't excite an immune reaction against our own cells. So our immune system is trained to recognize our own substances and not react against them, but will react against foreign substances. And that reaction by our immune system against foreign substances results in the production of antibody. And antibody and antigen will react together. If you've been following the whole thing about the COVID crisis, you'll realize this is tremendously important for us, because we are in taking a vaccination, we're trying to train our immune systems to react against viral proteins. So the same kind of system is going to start operating here. And have a look. On our red blood corpuscles, we have possibility of bearing various antigens. This is determined genetically. It depends on what your biological mother and your biological father had. But here are the possibilities. There is one antigen called A antigen. If a red blood corpuscle, if a person has red blood corpuscles which carry just the A antigen, then that person is said to be blood type A. Their own immune system will not react. It would be trained not to react to the A antigen. So A blood type A, there's no activity against A antigen. Similarly, there's another blood type called the B type. And blood type B has B antigens. It's a different molecule to this. I have a B antigen. Again, somebody with B type blood, their immune system will not react against B antigen. Other people can inherit both. They can inherit the genes for both A and B antigen and bear both on their red blood corpuscles. We say that they are type AB, and that both antigens are present. But then there are people who inherit no antigen genes at all. They are not able to produce antigen. Their red blood corpuscles are unlabeled with these antigens, and that we call type O. So there are our four major blood groups that you've probably already heard of: type A, type B, type AB, and type O. So this is the way that the system works. And this is, we're going to learn now how we can determine what blood group somebody belongs to. By the way, you should all know your blood group, and you should keep a record of your blood group somewhere, because in an emergency, it saves a lot of time if you are able to tell, if medical personnel, etc., are able to know your blood group beforehand. Everybody should know their blood group. So that plasma here, I told you it was full of proteins. Many of those proteins are antibody. They are antibody proteins, which our immune system has produced in an effort to protect us from infection. So we have circulating antibody that circulates in our blood in order to protect us from infection. It recognizes foreign antigen and reacts against it and neutralizes it, stops it from being able to work. So if our blood contains these antigens, we need to realize each of those antigens, very, very specific, antibodies rather, is very, very specific for a particular antigen. Antibodies are, the molecule itself is actually Y-shaped like that. And it has 2 reactive ends there and there. And it will only bind very specifically to a particular antigen. So it can bind an antigen there, and it can bind an antigen there. In other words, it can bind 2, right? So let's just imagine we've got a red blood corpuscle floating along, and it's got a antigen, and it encounters an anti-A antibody. It can stick there. Another red blood corpuscle coming along, it can come along and it can stick to the other one there. So we've got 2 red blood corpuscles stuck. Now we've got another antibody molecule comes in and sticks to the red blood corpuscles. Red blood corpuscles are covered with the antigens. They get covered with antibody. The antibodies stick to lots and lots and lots and lots and lots more red blood corpuscles. And very soon, very, very rapidly, we pull together so many red blood corpuscles that they actually come out of solution. We say that they agglutinate. They fall out of solution visibly. You can see it happening in front of you. And this is the test that we're going to use to determine a blood group. We're going to see whether blood corpuscles agglutinate with specific antibodies. We have the antibodies in a little, we purify them. We have them in little bottles as reagents that we can add to a blood sample and see whether the blood coagulates or not. So this is the way it works. Now, here's the logic that you've got to get into your heads. Well, first of all, let's just think about the blood type. Okay, group A, here's a red blood corpuscle. On the outside of the red blood corpuscle, it has the A antigen there. Okay, this person is group A. Are they going to produce anti-A antibody? No. No, that's their own blood group. That's their own proteins. They're not going to produce antibody to it. Instead, they produce antibody to B. B antigen is foreign to them, and they produce anti-B antibody. If you looked at their serum, it would be full of anti-B antibody. So group A protein, it's actually another kind of protein, but anyway, we'll just call it protein. That's the antigen, A antigen on the outside, right? The person produces not anti-A, but anti-B. And B would be foreign to this person. They produce anti-B. What about somebody with B group? Here's the B blood corpuscle. Here's the B antigens on the outside. What are they going to produce? They're not going to produce anti-B. They're going to produce anti-A. So they're producing anti-A. If they have the B antigen, they produce anti-A antibody. What about somebody with both, with A and B antigen on the outside? They're not going to produce any antibody at all because they're not going to produce anti-A, they've got A. They're not going to produce anti-B because they've got B. So they produce no antibody at all. But what about somebody who's group O? They have none of these antigens on the blood corpuscle. They produce both antibodies. They produce anti-A and anti-B. Okay, so it's just a trick. Just remember that you produce antibody to what you don't have. You produce antibody to what is not your own characteristic. So we're going to use this now. We're going to use solutions of these antibodies. And we're going to put them together with blood and see exactly what happens. When we do that, as I've described before, here, for example, this is A blood with A antigen. These are A blood corpuscles. I'm going to put in anti-B antibody, and look what it does. It starts to stick them all together. They begin to clump. That is what we call agglutination. So they begin to agglutinate, clump together. And they do so much that they actually come away from the serum and they become visible. And I'll just tell you there are other proteins and things that you can do the same thing with. But this AB system is the one which was first characterized. It's easy to do, it's a standard procedure, and it is still the most widely used of all the blood typing systems. So just remember the way that it works. That you produce antibody to the antigens you do not possess. So if you've got A, you produce anti-B. If you've got B, you produce anti-A. If you O and you've got none of them, you produce anti-A and anti-B. Now, I will just mention that this system here, this is another protein, which also reacts in the same way. I'll describe it in a second. This is called the RH system. So we just, for the moment, we're just going to think about the ABO part of the typing. We'll talk about the RH in a minute. So this is actually what it looks like. You take drops of blood, and you're going to add antibody. So I've got a drop of blood, and I'm going to add anti-A, a drop of anti-A antibody to this drop here, a drop of anti-B to this drop here. Look what happens. This is type AB blood we're testing. Does it have A antigen? Yes, it does. And we've added anti-A serum. It sticks all the blood corpuscles with A antigen, sticks them together, and they come out, become visible in these clumps. What about B antigen? Yes, it's AB, type AB. It's got both antigens. It's also got B. Add anti-B antibody, it will stick them together and they will agglutinate like this. This is actually what you see. It happens very, very quickly. It happens within 30 seconds. You see them clumping and falling out. Okay, so this person clearly is type AB. They react to both anti-A antibody and anti-B antibody. What about type B blood? Well, here's type B blood, and I add anti-A serum, anti-A antibody. There's no reaction. Why? Because this is type B blood. It has no A antigen for a reaction to occur. Add anti-B, and the same reaction happens. It agglutinates because it's got B antigen. Type A, same kind of logic, and it reacts to anti-A antibody, does not react to anti-B antibody. What about type O? It reacts to neither because it's got neither antigen. The antibody is testing for the presence of the antigen that it reacts against. Anti-A is testing, is A antigen present? If it is, it'll agglutinate. Anti-B is testing for the presence of B antigen. If it's present, it will agglutinate. So it will stick together. A, in this case, anti-A will stick together, and A type blood and AB. Same. B anti-B antigen, antibody, sorry, will stick together, will agglutinate AB blood and will agglutinate B blood. So this is that gives you a clear, simple, quick way of determining, is this A, B, AB, or O? So when we look at the population in general, about 43% of the population is type O, 42% is type A, 12% type B, 3% type AB. AB is the least common of all. And this, just to remind you, type A produces anti-B antibodies. Type B produces anti-A. Type AB doesn't produce any antibody to A or B. Type O produces both anti-A and anti-B. Remember something, when we're doing these tests here, we're artificially adding antibody. This anti-A and anti-B, we've got a little bottle of it. We add drops of it. This is not naturally occurring in the blood sample. Okay, so there is one other factor, blood factor, that is taken into account, And this is tests for the presence of another antigen. It's called antigen D. And if you have antigen D present on your blood corpuscles, then you are said to be Rh positive. So I think most of you have heard of that. So people with the antigen are Rh positive. If you don't have it, you're Rh negative. It's the same logic. If you've got the Rh, the D antigen, and you're Rh positive, you do not produce antibody to it. Otherwise, you'd be sticking together your own blood corpuscles. You produce the anti-D antibody if you are Rh negative. So the Rh is a little bit different because the vast majority of the population actually are Rh positive, 85%, in fact more usually, are Rh positive. Only about 15% of the population are Rh negative. With the same rule applying, we obviously have to take into account the Rh factor, especially in giving blood transfusions. This is the one place where it is really important because you do not want to give the wrong Rh to a recipient. What happens if you give the wrong blood group to a recipient is that the donated blood begins to get coagulated. So if, for example, if you are if you are A positive, that's your normal blood group, you are going to be producing anti-B antibodies, for example. So you get B blood infused into your own antibody will react against the donor blood and start to coagulate it, which is a very, very, very dangerous situation indeed, and can rapidly result in death. That is why blood typing is so very important. It's important that you receive blood that is as compatible as possible. And these are the major groupings which have been determined to be the most important for blood donor safety. A, B, AB, O, and then the Rh factor, positive or negative. Oh, there's one other circumstance. Hold on, I'll just explain it very, very briefly. It's not in detail at all. There is one circumstance where the Rh factor becomes very important, and that is it's possible for a woman who is Rh negative to carry a fetus that is Rh positive because they've inherited the gene from the father. So the fetus can be Rh positive, the woman Rh negative. The problem is that the woman can produce antibody to the Rh factor in circulating in the fetal blood. It's not so much a problem when in development in the uterus, but once the child is born, the maternal mother's circulating antibody in the child's blood can begin to coagulate the infant's blood. Nowadays, it used to be a tremendous problem. Nowadays, it's not a huge problem. It's been established well in advance the infant will be given a complete blood transfusion as soon as they are born to remove the mother's antibody, and the child will be on its way. But in the old days, if an Rh negative woman gave birth to a baby where this was happening, it produced the baby would begin to be deprived of oxygen, or all its blood corpuscles were being coagulated. The baby would turn blue, and this was referred to as the blue baby syndrome. Almost unheard of these days in any country that it has any reasonable medical structure. Okay, this is what a blood test card looks like. When you actually do the blood test, you put small drops of blood, and then you add the various antisera. Antisera, they're just solutions of specific antibody. And don't have a fit this whole diagram. Let me just move that out of the way. So this is what you'll actually see when you're doing this test. We're here in this case, what we're testing is A blood. And this, have a look and you'll see here we put a drop of blood and we add anti-A antibody. And as we've explained before, we get a coagulation. Why? Because the red blood corpuscles have A antibody on them. Add anti-B, nothing happens. Why? Because the blood corpuscles have no B antigen. This is A blood. Okay, so this is clearly group A. Here's the anti-D. This is the Rh factor. And we add anti-D, and you can see that it coagulates. That means the blood corpuscles have both A antigen and they have the D antigen. This is A group, Rh positive, and we say that is A positive. You always do a control. You always have a control that's done just with a little bit of serum with no antibody, because if you, it can happen that the blood can begin to coagulate itself out of solution, and then you toss that and you'd redo your test. So the control remains unagglutinated. So here are all the various groups, and this is how they would look if you did the test. And we won't go through them all, but I'll just give you a couple just to show you the basis of it. Let's look at O positive blood. What are we testing? We're going to test with anti-A antibody, anti-B antibody, and anti-D antibody, and we always have a control. So A positive, well, anti-A, is it going to react and agglutinate? No. Why? Because O blood has no A antigen. Anti-B, is it going to react to anti-B antibody? No, because it doesn't have a B antigen. The person doesn't have a B antigen, right? They're not going to be producing anti-B, and they're not going to be producing anti-A, right? They're not going to have a reaction to B or A. Anti-D, look what's happened. It is coagulated. That means it's got D antigen on the red blood corpuscle. So it is Rh positive. That is what it is. O reacts to neither anti-A nor anti-B. O positive because it has the D antigen. Control is fine. Your test is fine. Let's take another one. Let's take A, and we'll do A negative. It's not a very common blood group, but nonetheless, here's our blood drop. We added anti-A antibody, and it agglutinated. Tells us that those blood corpuscles have A antigen. We tested with anti-B. There's no agglutination. Why? Because this is A blood. It doesn't have a B antigen on the surface. We test now with the D, with anti-D antibody rather, no agglutination. That means that this person lacks the Rh factor. They are Rh negative. So this is A negative. The control is fine. Let's just have a look down here. This person's been working too late at night. They've left things too long. They've done something wrong, or their reagents are old or something else. They're testing blood, and they say, they see the A antigen, you've got agglutination. Anti-B antigen, testing for B antigen, they've got agglutination. D antigen, they've got agglutination. Huh. This, you could say that this person was AB positive. But can you really? Look at your control. No. You can't. This must be thrown out. This must be thrown out. You've got reaction on your control when you haven't added any antibody. So you don't know. You don't know if these agglutinations happen because they react against antibody or whether they just coagulated out. That's the role of the control. Have a look through here. These are all the possible blood groupings, and be ready. In test, you're going to be presented with one or two of these rows and asked to tell me what blood group they are. It's perfectly logical. Okay. If you test with anti-A and it reacts, it's got A. Test with anti-B and it reacts, it's got B. Test with anti-D and it reacts, you've got the D antigen. That is how the blood groups are determined. Okay, so this is for I see this is for Quebec, much the same all over the world. These are the proportions of the blood groups, so about 85% of the population are Rh positive, as I told you. About 15% of the population Rh negative. And here are for the different groups here. How many are O, A, B, and AB? All that I don't expect you to remember the exact percentages. What I do want you to remember is the least common is AB, and AB is also highly, highly desirable. Why? Well, because AB can receive blood from anybody. You can receive blood from O, A, and B because you don't produce any antibody to those blood groups. So this is called a universal acceptor. O blood is called a universal donor as long as the Rh factors are compatible. O is universal donor because it has their blood has no antigen on the outside for your antibodies to react against. But in forensic terms, determining the blood group at least allows you to say the person who dropped this blood, the person that this blood came from, belongs to a particular group. And we know the size of the group. So that is class evidence. It provides us with class evidence, but that class evidence may be very important. You realize if it was somebody with a very rare blood group, like somebody who is AB negative, heavens, that's a small group of the population. It becomes less significant, of course, when we talk about something very, very common, relatively common rather. The O positive, for example, is 39%. Nonetheless, it allows you to place the donor of that blood into a particular group. And that, as I told you, may be as important for excluding somebody as for including them. You have a crime scene where you have blood which you are fairly sure came from a perpetrator. Blood on a blood trail, for example, leading out. That has evidently come from a cut, from a wound or something. You test it, and it is O positive, right? Well, that puts that blood donor into a grouping, but you have a suspect, and you look at them, and they are B negative. What? They could not have donated that blood. They could not have produced that blood trail. So that may be as important as in being able to place them in a group, may be as important to exclude them from a group. Okay, so basically that is how the blood typings are done. Now let's have a talk about the actual antibody, because antibody itself is such an interesting substance. We can actually manipulate antibody. We can get animals to produce antibody for us. This is very often done using rabbits. If we inject substances into a rabbit, foreign substances into a rabbit, we can get the rabbit to produce antibodies to that substance. We can then take the rabbit blood, extract its serum, and extract the antibody from the serum and use it to test for the substance that we injected. Let's say we want to test for cocaine. Well, we can get rabbits to actually produce antibody to cocaine. And we can, just as we use the serum to test for the blood groups, we can use antibody then to test for the presence of cocaine, maybe in blood serum or in a body fluid or something like that. So This works on the principle that we described before when we saw the red blood corpuscles getting stuck together by the antibody. This is much the same. We're going to use something very like that. We're going to introduce a substance and see if antibody sticks to it sufficiently that it will actually precipitate out. We'll see it come out as out of solution as a precipitation. There are various other tricks that we can use as you'll hear, but that is the basic principle. Somewhere along the line, we're going to be testing for the binding of antibody to an antigen. So this is poor rabbits are a foundation in all of this, but we can actually commercially buy antibody, which has been produced for many, many different substances, but this is the basic way in which it's produced. We introduce the drug. We have to, there's a twist to the tail. We do usually have to attach the drug to a protein carrier that we inject into the rabbit, but the rabbit will actually produce antibody to the drug. So, very often they will produce several different antibodies which may stick at different parts of the drug. So when we extract the antibodies, what we're doing is we're actually extracting something of a mixture of antibodies, but they're all specific. They will be specific to this particular drug. So this is, when we do it that way, we call this a polyclonal antibody. It means that the poly tells you that there are many different kinds of antibody being produced, but they are specific for one particular antigen. They're just sticking to different small parts of the antigen. Okay. There is another way of producing antibody, which is called a monoclonal antibody process. It is described in some detail in your textbook. You can read through if you want, but I'm not expecting you to know the details of it. Monoclonal antibodies production is extremely laborious and very expensive, but I do want to mention it here again because of the current COVID pandemic. Monoclonal antibodies are being used in the treatment of COVID infection. Unfortunately, it's very difficult to produce monoclonal antibodies in great bulk. So they tend to be rare and rather expensive. Then it's not something which is routinely available from a pharmacy or something like that. But monoclonal antibodies are extremely effective and extremely specific. They to a particular antigen. So they can be a very powerful therapeutic mechanism. And this is what people are being given who are infected with COVID, especially if they look like they're going to become seriously ill. They may be given a monoclonal, very, very specific antibody. It's a single line of antibody, which interacts with only one specific part of the antigen. The normal cheap way of doing it, as I've described through the rabbits, that is a polyclonal. It works really, really well. It's cheap and it's very easy to do. So that is, polyclonal is very frequently used, not very effective therapeutically. Okay, so there are a number of different tests which are available, which are all based on assaying how well antibody interacts with a particular antigen and using that as a way of quantifying the presence of something and also of characterizing it, saying this is definitely what we have present in the blood and here it is present in this particular quantity. So this is just one of the techniques. There are many of these techniques. There are of these immunological assay techniques. There are many of them. Here's just one of them. It's called the EMIT technique, enzyme multiplied immunoassay technique, EMIT. And it's used specifically for examining for specific types of drugs, especially in urine. and attempting to quantify them. So, for example, I've already used the example of cocaine, so we'll just carry on with that. So let me just actually use the diagram because it's easier to explain. So what you usually do in an assay like this is we have antibody. This has been produced from the rabbit or whatever. So we have antibody, let's say it's antibody to cocaine. Very often what will be done is it'll be bound to a surface, like a little miniature petri dish or something. So there it is, that's the antibody there. So this is anti-cocaine antibody. And now we introduce we're going to introduce a urine sample. We're going to test a urine sample to see whether it has cocaine present in it, and we're going to try and determine a concentration. So this is the way it's done. Just for the moment, just realize that if cocaine is present, here's these dark things here. This is cocaine. It will stick to the antibody because it's going to react. It's going to coagulate with it like that. Here's the trick in this technique. What we're going to do is we're going to introduce a known quantity of cocaine, but that known quantity of cocaine has been specially treated. When it binds to the antibody here, it's got a marker on it, usually a dye, sometimes a fluorescent dye, sometimes a visible dye. The dye is invisible as long as the cocaine is free. This marked cocaine is free. When it binds to the antibody here, it becomes visible. So it turns a color. Okay. So let's just say here we've got our antibody. We're going to here's our labeled drug. This is we're introducing artificially, the labeled cocaine. This person whose urine's being tested, they have no cocaine in the urine at all. What will happen is all of the labeled cocaine will stick to the antibody. This is going to turn a dark color, a relatively dark color. So now we're going to do the same thing, but we're going to do it with somebody who has cocaine present in their urine. So now we've got a mixture. We've got our artificially known quantity of labeled cocaine, and we've got an unknown quantity of free cocaine that is present because the person had been using cocaine. What is going to happen? In this instance here, there's going to be competition. There's going to be a competition between our artificially labeled cocaine and the free cocaine that's there because the person's been excreting cocaine. Okay, so they're going to compete for antibody sites. This is a permanent binding. If you bind one molecule here, another molecule can't come in and sit here. So now what's going to happen? Well, some of the antibody is going to bind our artificially labeled cocaine, but some of it is going to bind the free cocaine molecules. And what's going to, in the end, when we can wash out all the stuff and look, we're going to look at the surface. How many of these molecules have the bound labeled cocaine? How many have bound the free cocaine? This was very dark because these were all our labels. This is going to be much lighter. This is going to be much lighter because so many of the antibody molecules have bound to cocaine, which is unlabeled. It was the cocaine that was excreted. It has no label on it. We can measure the color. We can measure this color. We can measure it with the spectrophotometer, which is an instrument which measures wavelengths of light, the strength of wavelengths of light. It can measure, let's say this turns blue. We can measure with the spectrophotometer how blue this is and how blue this is. And because we know the quantity of label that we added, we can work out the actual concentration of free cocaine molecules. We've done two things in this test. The first is we've identified cocaine as being present in the urine. We've said this person has cocaine in the urine. Second of all, this person has cocaine in a particular quantity. It allows us to quantify the amount of cocaine that is present. In one test, we've performed two very difficult and very useful things. Okay. The reason we can say that cocaine is present because only cocaine will stick to these antibodies. Marijuana is not going to stick there. Neither is heroin. We can have separate tests for marijuana and for heroin. If you wanted to do that, you may well do that. But if cocaine was present, this gives you a positive for cocaine, and it allows you to compute how much cocaine is present in the urine. Okay, so with that said, realize that that's just one example, okay? Those immunological assays, there are many, many of them for all sorts of different things, and there are many different methods as well. That's just one, the IMIT is just one example for you of the way it could be used. Okay, so let's just say, let's go back to a crime scene. First of all, we look around and there are drops of a dark substance, perhaps on a sheet, perhaps on a floor somewhere, something like that. We cannot simply assume that what we're looking at is blood. There are many, many things which look like blood. So first of all, is it blood? Second of all, many things bleed. Animals bleed. Blood can originate from a cut of meat that you take out of the fridge when it's thawed. Blood could originate from a pet. There are all sorts of origins so we need to know, is it blood? And then whose blood? Is it human blood? If it's human blood, whose blood? Can we identify it to a person? Or we've already seen that using blood groupings, we can assign it to broad groupings. We can get a class of individuals that blood could have come from. But blood may also offer us the opportunity for individual identification if DNA is present. So blood has assumed an increasing importance in forensic investigations, especially as DNA analysis techniques have been developed. So let's go through those questions one by one. Is it blood? Well, this is a very old test. This was developed early on I think in the late 1800s, early 1900s. It's called the Kastle-Meyer test. And it uses a reagent, phenolphthalein, under specific circumstances. Phenolphthalein will react with hemoglobin. And so if you have a knife, do you assume that is blood? Well, you shouldn't. If you're a good forensic scientist, you don't just assume that it's blood and proceed. You test it to see if it's blood. And you use a Kastle-Meyer to determine, first of all, is it blood? Does it have a very high hemoglobin content? All you have to do, take a small sample. It can be very, very small. This is a very sensitive test. And then you test it with Kastle-Meyer reagents, which is a simple set of drops that you add to your swab. This is what a negative looks like. You should always do this. You should always have one swab that just has water or something on it to make sure that your reagents are working properly. So if you get a negative control, this is what a positive looks like. And this tells you this is blood. Okay, that's all it tells you. It tells you this is blood. Well, it doesn't even really tell you that, but it tells you you are dealing with a substance that is very, very, very high in hemoglobin, and that is almost certainly blood. Remembering also that blood can be of different origins. Blood which is in circulation we refer to as peripheral blood. So that is the blood that's would most commonly be encountered at a scene of violence, whatever. But there's also menstrual blood. That is a whole different topic because it's sometimes becomes very difficult to distinguish menstrual blood from peripheral blood, and yet it's a very important distinction to make. Nonetheless, menstrual blood will also give you a positive in this case. The investigator just needs to be aware that menstrual blood could be present under certain circumstances. So that's one way, the Kastle-Meyer, very old test. Here's another. This is a test which is quite frequently used to identify blood, especially in circumstances where an attempt has been made to clean. It is actually extremely difficult to clean up blood. The reason is the blood the blood corpuscles come suspended in that serum. And the serum is full of proteins, and the proteins, when they dry, are almost like a glue. And in fact, they don't even have to dry. When they encounter a surface and coagulation of the blood starts, they actually begin to bind to surfaces. And somebody attempting to clean blood, they're very seldom able to remove all of the blood. It may visibly be clean, but they leave behind traces of blood. And commonly nowadays, substances are used which react with the blood and produce a fluorescence, especially when viewed under ultraviolet light. The commonest of these is luminol. And luminol is extremely sensitive substance. It can detect blood that's been diluted 100,000 times. They can have washed a carpet, washed a surface over and over and over again. And if there are traces of blood, the luminol will show up. Luminol is usually sprayed onto the surfaces and is then examined under ultraviolet light. Photographs are taken very rapidly because the glowing, the fluorescence, fades after a while. So the lights are switched on, the ultraviolet lights are switched on, photographs are taken very carefully so that the size and the distribution of the glowing patches can be identified. So this is what the kind of result that you get. This is a carpet and a surface that have been cleaned. There is no visible blood at all, but spray it with luminol, and this is what you got. You've got a huge patch of blood there. You've also got on the surface here, there is blood. All of this is evidentiary. The pattern and distribution of the blood could be very, very important. It looks, for example, as if the body lay here and then was carried or dragged or something like that off in this direction here. So this is a typical luminol result. Here's another one. Again, this is a real crime scene. Something really bad has happened here. There's blood being spilt all over the sofa here. And then there's a clear drag trail out here to show exactly, it tells you a whole story right there. This might have been, I don't have a before picture, but it is quite possible that this had been cleaned up to one extent or another. Often people will do things like bring in a carpet cleaner or something like that, actually scrub carpets. It makes little difference. There are some disadvantages to the luminol, and that is that commonly used cleaning products may, in fact, also react. Bleaches, if the bleach is still present, and on the surfaces, if they're still damp with bleach, that sort of thing, it can also produce a reaction. So it's not infallible, but it can give extremely, extremely powerful visual evidence of pattern of a crime. Okay, so there, we've identified this blood. Is this human blood? The luminol tells us this is blood, but it does not tell us whether this is human blood. But human blood, in fact, does have particular characteristics, and those characteristics are carried largely in components of the serum of the blood, and on things like the proteins on the surfaces of the red blood corpuscles, as we've discussed before. If we take human blood and we inject it into a rabbit, the rabbit will produce a whole mess of antibodies specific to human blood. And it's, in other words, producing polyclonal antibody to human blood. And we can then extract those antibodies from the rabbit, and we can use it to test a sample of unknown blood to see whether it is human blood or not. If it is, it will produce a precipitation of one sort or another, just as we've described before. So this is referred to as a precipitin test. And it uses antisera, which are derived from an animal source, usually rabbits. Again, you don't have to do the rabbit thing yourself. You can usually buy the antisera from a manufacturer who produces it. There are also antisera to all sorts of other things. Antisera to dog blood, to cat blood, to cattle, pigs, etc., etc., etc. In case you need to carry on tests, you get a negative for your human blood. Well, then why is this blood there? What kind of blood is it? Oh, it's a pig. Somebody's been nutty enough to slaughter a pig in their lounge. Things happen, but it's not necessarily a crime. Okay, so it's the same idea as we talked about before. We get a human blood sample, put it into rabbits. They produce antibody to the human proteins, and we are then going to use the rabbit serum, the rabbit antibodies, and we're going to test our unknown sample to see if we have a reaction. And if we have a reaction, it tells us pretty certain that that is in fact human blood. So this is a classic precipitin test. This is a precipitin test done in a test tube. This would be particularly valid if you had a fairly large sample that you could test. And they will take a sample, make a solution. Let's say it'll work even with dried blood, work with wet blood as well. But you would make a solution in a serum, and perhaps filter it, and layer it on top here above a layer of the antibodies. So this is rabbit antibody down here, and on top of it you layer your unknown sample there. Is this human blood or not? The antibodies in the rabbit serum are against human blood. If they react, they're going to agglutinate there. They're going to make a precipitate in this zone here. And here you can see it visible here. That's why it's called the precipitin test, because it precipitates out. Here's our unknown sample above. Here's the rabbit serum below. And here in the middle, it's reacting against the human blood proteins and is producing a visible layer of precipitation. That is the precipitin test. This is a positive for human blood. So this assumes that you've got quite a large sample. It doesn't have to be very big, but a fairly large sample. If you've got a very small sample, it's also very much more convenient. You can just use a very little. You can do this in a gel. It's basically like a Jell-O matrix, okay, that you cast in a bed. It's just and you cut wells in it, and in one well you put your antigen, in the other well you put your antibody, and you put an electrical field across. And in actual fact, the antigen and antibody will move in opposite directions. Antigen moves one way, antibody will move the other way, and where they meet here, you get a precipitation if it's a positive. You've got anti-human antibody, and you've got an unknown. You want to know, is this human or not? If you get this precipitation in the middle, it's the same idea. It's a precipitation, it's still a precipitin test. This is very advantageous though, because you can use very small quantities. This is very simple to do, and it's a routine method of doing it. Okay, so then we've said it's blood. We did Kastle-Meyer or luminol or something. We said it's human blood, but whose blood? Well, this is an entirely different story, because the blood groups are only going to give us a class evidence. They're not going to allow us to individualize a blood stain or whatever to a particular person. But remember that thin layer of white blood cells. because those white blood cells contain DNA. And DNA was going to allow us to actually individuate blood. We can tag a particular blood stain to a particular person if we can extract enough DNA and we can characterize that DNA. And that is why blood stain has assumed increasing importance in forensic investigation. But we'll talk about DNA analysis in another set of lectures, not in this one here. So let me just quickly also describe a couple of the other body fluids that we need to talk about. The first is saliva. Saliva is very important because our saliva contains cells. The insides of our mouths are coated with a mucous membrane, which reproduces itself very, very rapidly. The cells of our mucous membranes are extremely loose, and saliva contains cells from our mucous membranes. The cells contain DNA, so saliva can be an important source of DNA. There are one or two other things which might be looked at in saliva, but they often saliva is, you see saliva as presumptive. You may see something which you're not entirely sure is the saliva or not. I'll give you an example. Any instance where there is a bite mark, the surroundings around the bite mark might be tested for saliva. And one of the ways in which you test whether or not substance is saliva is to test for an enzyme called amylase. Our saliva is very rich in amylase. It's the very first of our digestive enzymes, and it breaks down starch. It breaks down starch to smaller sugars and glucose. And you can here, these are tests for amylase. This is a fairly weak. This is a stronger test for amylase, informing you that in fact some stain was in fact saliva. You may also want to test to distinguish saliva, what looks like a stain, which could be saliva or could be some other body fluid like semen, for example. You may, amylase is not present in semen. So semen is another really important body fluid for various reasons. First of all, obviously, it is commonly associated with sexual assault as a result of ejaculation by the perpetrator. Semen is also very important because it is very, very rich in DNA, and so that it can be tied to an individual. Semen can also be used, by the way, to determine a blood group. And in the old days, before DNA analysis, that was one of the few things that could be done with semen, was that it could be tied only to a class of suspects. It couldn't be individuated. Nowadays, semen is of particular importance because it is such a rich source of DNA that you only need a very, very little in order to perform the analysis. So again, this is you may have stains of many different origins which resemble semen. So the first thing to do is to determine, is this in fact semen or not? And the classic test is to test for an enzyme which is present in high quantities in semen. It's produced by the seminal vesicles, and it's called acid phosphatase. And you test for acid phosphatase with a simple acid phosphatase test, and it produces this distinctive blue coloring. This is a sheet or something before it was tested. This is the coloration for it. And this can be done on a very, very small amount. So you can test the stain directly, or else you can take a swab it, and then test the swab as well. It is a sensitive test. So that is a presumptive. If you get a positive alkaline phosphatase, you can presume that is this semen and proceed collect and everything. If you get negatives, then it could be something else. They could have spilled their tea or whatever. But if it's presumptive for semen, you would then proceed to collect properly and then proceed with further analysis to define whether or not it is semen. And semen is definitively identified by the presence of spermatozoa. Here, this is what human sperm look like when they're stained under the microscope from such a smear. Clearly identifiable with the head and with the tails behind. The structure of sperm, etc., gives you no information. One person looks exactly the same as another person. But each of the sperm contains DNA, and that DNA can now be analyzed and it can be individuated. There's another protein which is specific to seminal plasma, which is called P30. Just all that you need to remember is if we see specific semen proteins and identify them, or if we see spermatozoa, it tells you this is definitely semen. Okay, so these are obviously very important in cases of sexual assault and especially in cases of rape. If the first obvious thing is in a case of rape, the victim is very, very carefully examined. Unfortunately, this in itself can be a traumatizing event, re-traumatizing event. But unfortunately, evidence, it's almost as if the rape victim becomes an object of evidence themselves, because there can be body fluids, hair, all sorts of things all over the victim of an assault like that. And that evidence does need to be gathered, and if it's going to be gathered, it's got to be identified. Can only be identified by careful examination. It's in itself needs to be done with enormous sensitivity as much as possible, but in itself, I think probably rather a brutal thing to have to undergo, even if it has a hopefully helpful outcome. But suspects must also be very carefully examined, because especially within a reasonable time period, 24 hours or so, it may be possible to find the victim's DNA on them. When an assault like that occurs, the perpetrator acquires DNA as well as donating it. Remember Locard's principle. Every criminal leaves something and takes something away. This is much the same. So perpetrator is very carefully examined to see, do we have any victims' body fluids, victims' DNA, victims' hair. In order to do so, they also have to be very carefully examined, and the clothing has to be very carefully examined. But it may in fact produce critical evidence, particularly if it's done relatively shortly after. There's just one thing to point out as well, and that is in a case of rape, of vaginal and rectal rape, deposition of semen, the semen may actually last for some time, and it may be evidentiary for quite a long time after the assault. And so, for example, just have a look through here, and you'll see that some of the specific enzymes and things associated with semen, well, they do last, but they don't necessarily have that much evidentiary value. But look at the sperm. Sperm will survive mobile with their tails. It says 4 to 6 hours, but I believe it actually may even be longer than that. This is probably a median. And then sperm themselves, the sperm cells themselves, can last 3 to 6 days. That may be extremely important because they are a source of DNA. So the sperm itself is can be a very, very valuable source of forensic information. Look back, when we discussed about collecting biological evidence. And remember that biological evidence has to be very carefully collected. We need to avoid any contamination by the examiner, and it needs to be kept as in conditions where bacteria, fungi, etc., don't grow and contaminate the sample. So they're packaged carefully, packaged separately, packaged in paper bags, not in plastic, which encourages fungal growth. And biological samples like this would usually be kept under refrigerated conditions until they are sent to the laboratory. Okay, I'm a little bit over time, I'm sorry, but that concludes that for today. And I'll see you on Friday.